I want you to imagine, just for a second, trying to raise one of the world's most important sources of high-quality animal protein.
Right. Broiler chickens.
Exactly, broiler chickens.
Right.
But imagine trying to raise them in literally one of the absolute harshest, most unforgiving environments on Earth.
It's a completely different ballgame up there.
It really is. And today, that is exactly what we are looking at. Welcome to our deep dive into a really fascinating scientific research article.
Yeah, the paper is titled Ramnoids as Novel Phytogenic Feed Additive for Broiler Chickens at High Altitude Cold Desert.
Which is a mouthful, I know.
It is, yeah.
But our mission today is to unpack this study because, well, why does this even matter to you listening? Poultry farming isn't just about, getting food from a local farm.
No, not at all.
It's his massive, critical pillar of global food security. I mean, nutritional health, rural livelihoods, it all depends on it.
And broiler chickens are actually one of the fastest growing sources of high-quality animal protein worldwide.
Right. We rely on them globally. So maintaining optimum poultry production is huge.
But doing that becomes incredibly challenging in extreme environments.
Which brings us to the setting of this study. The researchers focused on the Trans-Himalayan region of Leh Ladakh.
Yeah, and to really understand the study, we need to talk about that environment.
Because it is brutal. I mean, we are talking about an elevation of roughly 3,500 meters above mean sea level.
That's about 11,500 feet.
Yeah, you're practically in the clouds. And it's a cold desert. So you have these extreme temperature swings.
Oh, absolutely wild swings.
Right, like plus 35 degrees Celsius in the summer and then plunging down to a bone-chilling minus 35 degrees Celsius in the winter.
Plus, you have intense ultraviolet radiation because the atmosphere is so thin.
Right, and low humidity, harsh climatic fluctuations, basically everything a farm animal hates.
Exactly. But the core problem, the real invisible killer up there, is something called hypobaric hypoxia.
Hypobaric hypoxia. Break that down for us.
Well, it basically means there's extremely low oxygen availability because of the low atmospheric pressure.
So the air is just too thin to breathe properly.
Right. And when you put a broiler chicken, an animal that is genetically bred for rapid growth and high metabolic output, into that environment, the results are, well, they're disastrous.
The bird's entire system basically starts choking.
Yeah, these environmental stressors fundamentally impair their growth, their immunity, their metabolism.
The overall health just tanks.
Exactly. Which brings us to the microscopic level. The real invisible threat here is oxidative stress.
And this is where it gets a little counterintuitive, right?
It does, yeah.
Because you would think, okay, there is less oxygen in the air, so there should be less oxidation happening in the body.
Right. That sounds logical. But it's actually the opposite. To understand why oxidative stress becomes the major concern at high altitude, we have to look at the mitochondria.
The powerhouses of the cell.
Exactly. The mitochondria rely on this steady, predictable flow of oxygen to generate energy. They use a mechanism called the electron transport chain.
Okay.
But when the partial pressure of oxygen is drastically reduced at high altitude, that entire transport mechanism gets disrupted.
It sort of chokes up.
Yeah, it starts to severely malfunction. And because of that malfunction, it leads to the excessive production of reactive oxygen species, or ROS.
Which most people probably know as free radicals.
Right. Free radicals. These are highly unstable, very reactive molecules.
Because they're missing an electron, right?
Exactly. They are desperate to steal an electron from anywhere they can.
So they basically go on a rampage.
They really do. They trigger these destructive chain reactions inside the cell. They start damaging the cellular DNA, warping proteins, and attacking lipids.
Okay, I want to jump in here with an analogy because I think this can get a little abstract.
Sure, good.
So if we think of a single cell as like a wooden cabin.
Okay, wooden cabin.
And the mitochondria are the fireplace inside that cabin, right? Providing energy and heat.
That's a great way to look at it.
Under normal conditions, down at sea level, the fire burns clean. But when you choke off the oxygen supply up in the Himalayas, that fire starts sputtering and popping.
Right, it gets chaotic.
And those free radicals are basically rogue, burning sparks that shoot out of the fireplace and land on the wooden floor of the cabin.
I love that analogy. That is exactly what happens. And the damage to the lipids, that's the wood floor catching fire. We call it lipid peroxidation.
Which literally burns the cell membranes.
It destroys them. And when enough of these cellular cabins catch fire, the bird's overall growth and immune competence just collapse.
So to stop this, the body needs a team of natural firefighters.
Yes, antioxidants. Their job is to extinguish those sparks before they burn the house down.
But the chickens up there are so stressed they don't have enough firefighters on their own.
Exactly. And historically, the agricultural industry tried to fix this by pumping the birds full of synthetic growth promoters and antibiotics.
Which, we are trying to move away from globally.
Right. There is a huge push for natural alternatives, and that is where phytogenic feed additives come into play.
Okay, let's unpack this. Phytogenic feed additives.
Right. So phytogenic additives are basically natural plant-derived products,
Okay.
and they are rich in bioactive compounds that enhance digestion, improve immunity, and naturally reduce that oxidative stress we were just talking about.
So they promote healthy growth without relying on synthetic drugs.
Exactly. And the star of this particular study is a plant called rhamnoids.
Commonly known as sea buckthorn.
Sea buckthorn, yes. It's this really remarkable medicinal shrub.
And the fascinating thing about this plant is where it grows. It naturally thrives in the exact same harsh Himalayan environment.
Right, at elevations of 3,000 to 4,500 meters.
So it's sitting out there in the freezing cold, blasted by UV radiation, with virtually no oxygen.
And it survives. In fact, it does more than survive. Every single part of this plant, the fruits, leaves, seeds, berries, is just loaded with valuable phytochemicals.
We're talking polyphenols, flavonoids, carotenoids.
Vitamins, amino acids, polyunsaturated fatty acids. It's a biochemical fortress.
And what's really cool is that this isn't some brand new laboratory discovery.
No, not at all. Sea buckthorn has been heavily utilized in traditional medicine for centuries.
Specifically, the traditional Himalayan Amchi system, right?
Right, the Amchi system, also known as Soa Rigpa. Traditional healers have used this plant for a very long time to treat ailments and combat environmental stress.
So the researchers here basically looked at this ancient botanical wisdom and decided to put it to a rigorous scientific test.
Exactly. The objective of the study was to see if an aqueous extract, a water-based extract of the sea buckthorn fruit, could actually improve the survivability and health of these high-altitude broiler chickens.
And do it economically.
Right, that's key. But before they could just start giving this extract to a whole flock of live birds, they had to prove it actually worked in a lab.
You can't just throw a shrub into the water supply and hope for the best.
Exactly. So they started with in vitro laboratory experiments.
To test the firefighters in a petri dish.
Yeah. They had to evaluate the extract's antioxidant capacity. And to do that, they used some very specific standardized preliminary tests.
Okay.
They used the FRAP assay, the DPPH-free radical scavenging assay, and the ABTS radical scavenging assay.
Whoa. Okay, hold on. Let me jump in here. That is a lot of acronyms.
Yeah, it's a bit of alphabet soup.
Let's break those down for the listener. What are these tests actually doing in simple terms?
Fair enough. So FRP, DPPH, and ABTS are just different standard laboratory methods used to measure how strong an antioxidant is.
Okay, so it's a stress test for our firefighters.
Exactly. We know how well a standard antioxidant, like vitamin C, ascorbic acid, fights off free radicals. So we use these tests to see how effectively the plant extract scavenges or neutralizes those same free radicals compared to that known standard.
I see. It's basically comparing the sea buckthorn to the gold standard of firefighters.
Exactly, and that the lab results were incredibly promising. The extract showed exceptionally high concentrations of polyphenols, like gallic acid equivalents and flavonoids.
So it's packed with the good stuff.
It is, but they didn't stop at just chemical tests. They also ran a really fascinating immune stress test.
Right, using live chicken cells.
Yeah, they cultured chicken white blood cells, specifically lymphocytes.
Okay.
And then they intentionally exposed those cells to toxic hydrogen peroxide, or H2O2.
Which basically simulates severe oxidative stress, like dropping a chemical bomb on the cells to mimic being at 11,500 feet.
That's a great way to put it. And the results were amazing. The sea buckthorn extract not only protected the cells from dying off, but it actually stimulated the immune cells to proliferate.
Wow. So it didn't just hand out fire extinguishers to the existing cells, it actively recruited more firefighters to the squad.
Exactly. It boosted the immune response in the dish.
Which is incredible. Yeah. But, as we know, a petri dish doesn't have a beating heart or a digestive system.
No, it doesn't.
So with the cellular firefighters proven to work, the researchers transitioned to the real-world in vivo experiment.
Right. The 42-day field test.
Let's outline how they set this up, because it was a pretty rigorous design.
It was. They took 105 one-day-old Rhode Island red crossbred broiler chicks.
And they placed them in a specialized solar poultry house to deal with the extreme cold.
Yes. And they divided these chicks into a control group, which just received a standard basal diet,
Okay.
and then 6 treatment groups. And these treatment groups received different concentrations of the sea buckthorn extract in their drinking water.
Ranging from 100 milligrams per kilogram of body weight all the way up to 800 milligrams.
Right.
And this is where I have a specific question. Why give them 6 different doses ranging all the way up to 800? I mean, if this stuff is natural and healthy, shouldn't more just automatically be better?
If we connect this to the bigger picture, it actually comes down to how biological systems process substances. In biology, there's almost always a Goldilocks zone.
Right. Not too hot, not too cold?
Exactly. Too little of the supplement, and it's ineffective. It won't neutralize the oxidative stress.
Right.
But too much isn't just a waste of resources. It can actually cause unintended metabolic imbalances.
Because the liver and kidneys have to work overtime to process all those phytochemicals.
Precisely. If the bird is expending extra energy just metabolizing a massive dose of the supplement, it's taking energy away from growth. So evaluating multiple dose levels helps the researchers find that optimum supplementation strategy.
Maximum benefit for the lowest metabolic and financial cost.
Exactly.
Okay, so that builds the anticipation. We have the 42-day trial, the extreme environment, the 6 different doses. What actually happened?
Well, the data was very clear. Treatment group T3 was the undeniable winner across almost all the metrics.
And T3 was the group receiving 200 milligrams per kilogram.
Right. The 200 milligram dose was the Goldilocks zone.
Let's get into the specifics of how they thrived, starting with their physical growth.
So physiologically, the improvements in the T3 group were significant. By the end of the 42 days, they had the highest body weight. Okay. And crucially, they had the best feed conversion ratio.
Which means they were digesting their food more efficiently and turning it into muscle better than any other group.
Yes. But to really understand why they grew better, we have to look at their blood work.
What did the blood show?
The T3 group showed significantly increased levels of total protein and globulin in their blood.
And higher total protein indicates higher protein synthesis, right?
Right.
Like the bird is literally able to build more muscle.
Exactly. And higher globulin levels indicate a boosted immune system.
So they were bigger and they were healthier.
They were. And there was another interesting finding in the blood work regarding cholesterol.
Oh, really?
Yeah. The birds in the T3 group had lower levels of LDL, which is the bad cholesterol.
Okay.
And higher levels of HDL, the good cholesterol.
That's fascinating. You usually hear about cholesterol with human heart health, but it matters for chickens too.
It does. It's a great indicator of overall metabolic health. But the most important blood marker they looked at connects right back to our earlier discussion about oxidative stress.
Right, the fireplace sparks. Did the extract actually stop the cellular damage?
It did. They tested the blood for a marker called MDA.
Malondialdehyde.
Right. MDA is a marker of lipid peroxidation.
So going back to our analogy, if the free radicals are the sparks and the damaged cell membrane is the burning wood.
Then MDA is the ash left behind. It's the physical proof of cellular damage.
Got it.
And the blood of the sea buckthorn-fed chickens, specifically our T3 group, shows significantly lower levels of MDA.
Wow.
And they had a much higher total antioxidant capacity overall.
The firefighters were definitely working.
They absolutely were. The extract successfully intercepted the free radicals.
And this all sounds great on a microscopic level, but how did that translate to the birds actually surviving the 42 days?
This is where the numbers get really stark. Let's look at the mortality rates.
Nah.
The control group, the chicks that received no plant extract, suffered a 26.67% mortality rate.
Wow, so over a quarter of the flock just died.
Yeah. And they were dying primarily from high-altitude conditions. One of the main culprits was ascites.
Ascites, that's directly related to the thin air, right?
Yes. Because there is so little oxygen, the bird's heart has to pump furiously to try and oxygenate the body. This creates severe high blood pressure in the lungs.
And eventually the heart just gives out.
Right. Ventricular failure. Fluid fills the abdomen and the bird dies. It's a huge problem at high altitudes.
And the other big killer in the control group was coccidiosis, right?
Yes, which is an intestinal disease. It tears through a flock when their immune systems are compromised by stress.
So the control group loses over 26% of its birds. What about our winning T3 group?
The mortality rate for the T3 group, the ones getting the 200 milligram dose, was slashed down to just 6.67%.
That is a massive difference.
It is. But here is the most incredible part. Out of that 6.67%, there were 0 deaths from ascites.
Wait, really? 0?
0. and 0 deaths from coccidiosis.
Oh, wow. So the sea buckthorn extract essentially shielded them entirely from the two biggest environmental killers.
Exactly. It protected their cardiovascular system and kept their immune system strong enough to fight off the intestinal disease.
That is honestly staggering. But of course, in farming, it always comes down to the bottom line.
Rate the economic.
If the plant extract costs a fortune to produce, farmers won't use it no matter how well it works.
True. But the researchers factored all of that in. They calculated the cost of the chicks, the feed, and the cost of producing the aqueous extract.
And
Even when factoring in the cost of the additive, the T3 group yielded the highest net economic return for the farmer.
because more birds survived, and the ones that survived grew larger using less feed.
Exactly. It's a win-win.
So what does this all mean? Summarizing this for you listening, we are looking at a major modern agricultural challenge. How do we sustainably feed a growing world in increasingly difficult environments?
And the answer here wasn't a new synthetic chemical.
No, it was combining ancient traditional botanical knowledge with rigorous modern science. A simple water-based extract of sea buckthorn at just 200 milligrams per kilogram allowed these vulnerable birds to genuinely thrive in a hypoxic, freezing desert.
It's a highly effective natural solution.
It really is. And I want to end today's deep dive with a final, slightly provocative thought for you to ponder.
Okay, let's hear it.
Think about this shrub, Hippopharamnoids. It evolved over thousands of years to survive the freezing, UV-blasted, low-oxygen extremes of the Himalayas.
Right, it had to adapt or die.
Exactly. And because of that brutal evolutionary pressure, it developed the exact phytochemical blueprints needed to protect a completely different, highly vulnerable animal placed in that same environment.
It's nature providing the exact tool for the job.
Yeah. So as our global climates become more volatile, as we see extreme weather patterns disrupting traditional farming, and as we are forced to produce food in harsher conditions.
What else is out there?
Exactly. What other seemingly harsh, extreme survival plants are just sitting out there in the wild right now? What other resilient weeds or thorny shrubs are secretly holding the biochemical blueprints we are going to need to adapt our global feed systems to a changing planet?
That is a fascinating way to look at it. There is probably a whole pharmacy of solutions out there we just haven't tested yet.
I think there is. Nature has already done the millions of years of R&D. We just have to be smart enough to look.
Well said.
Thanks for taking this deep dive with us today.